Hello everybody. In this lecture, I'll talk about the immunity. We'll talk about different types of immunity, and then we'll talk about three lines of defense given by the immunity. Then we'll talk about the complement system, inflammation, different types of T and B cells, and antibodies. So first question is, what do you mean by immunity? Immunity is the ability to fight the antigens. Your body has the ability to fight the antigens to protect your body. So that ability is the immunity. Now, immunity can be two different types. First, we divide the immunity into innate and adaptive. Adaptive immunity is also known as acquired immunity. Okay, so first we classify the immunity into innate and adaptive. Innate immunity we get from our birth. When we born, we get that immunity. So everybody gets it. Adaptive immunity is produced in your body in response to antigen. After your birth, you get exposed to many different types of antigens throughout the life, and your body produces the immunity against those specific antigens, right? So innate is fixed. Everybody has the innate immunity and we get by birth, but adaptive could be different in different person. If you got exposed to swine flu, then your body has that adaptive immunity against that swine flu. If someone else didn't get swine flu but got COVID, that person has adaptive immunity against COVID. So you see, the adaptive immunity depends on the exposure. Your body got exposed to what kind of antigen, and based on that the adaptive immunity develops or grows in your body. So it is highly specific, but innate is very generalized. So first, let me tell you the differences between the adaptive and innate immunity. So immunity is the ability to fight the antigens, right, to protect your body. First, we divide the immunity into innate and adaptive. This classification would be very helpful. Adaptive is also called acquired immunity, right? Now, innate immunity gives two lines of defense, the first line and the second line of defense. The first line and the second line. And adaptive immunity gives the third line of defense. Okay, so you can see three lines of defense. First line of defense is the covering and linings of the body. When something tries to enter into your body, for example any bacteria or virus, you know that your skin, which covers the whole body, right, tries to stop it. So skin, and if that antigen tries to enter through the nose or through the mouth, right, uh, inside the nose and your respiratory tract, for example, this is your trachea, you know that it is like a tube, and the inner lining, if I make a cross section, the trachea will be like this, right? So this is the lumen, and here you have the epithelial lining. Same thing you have in your gastrointestinal tract. So respiratory tract and gastrointestinal tract has inner lining, and mucus is secreted here, you know that. So you have mucus and inner lining, and that would prevent the microorganism or antigen from entering into your body. So skin is the covering of the body as well as the linings, right? Linings of the respiratory tract and the GI tract. If the antigen tries to enter through the food, through the mouth, then in the wall of your GI tract, inner surface has the epithelial lining as well as the mucus that is secreted will stop that antigen, right? So inner linings of the respiratory and GI tract. Makes sense? And we all get these things by birth. Everybody, right? Uh, so everybody has these structures to protect the body. Now, if this is your skin and somehow that antigen can pass through the skin or the lining, then what will happen? Under the skin you have the tissue, right? For example, you have the muscle tissue. So this is the tissue under the skin, and if the skin and mucous membrane and lining of your GI tract and respiratory tract fails to stop that antigen, for example, this is the bacteria, then it will enter into the body, right? And in your tissue you have some components that will now try to destroy this antigen. So in your tissue you have what? You have phagocytic cells or phagocytes. They try to engulf the antigens, right? So phagocytes or phagocytic cells, for example, macrophages, and also tissue secretes some chemicals. Those chemicals can destroy the antigens. Okay, so we can say that these belong to the second line of defense, because your first line was not able to stop the antigen, right? First line failed. So the antigen now has entered into the tissue, and in your tissue you have the second line of defense that includes tissue phagocytes and chemicals called the tissue proteins. That includes cytokines, interferons, these are the chemicals, okay, and complements, these are the proteins, tissue proteins, and they would try to destroy the antigens. So all these belong to the second line of defense. Now The third line of defense is given by the adaptive immunity. That means your body will produce antibodies or the lymphocytes that will try to destroy the antigens. So third line is the final step that would be given by the lymphocytes. Blood has the lymphocytes, one type of white blood cells, right? So lymphocytes would give the third line of defense. Now the lymphocytes could be two types: T cells or T lymphocytes and B cells or B lymphocytes. In my last lecture I mentioned that they fight differently. They know different techniques to fight the antigens. So T cell mediated or T lymphocyte mediated, and B cell or B lymphocyte mediated immunity, and those belong to the third line of defense. We'll talk about T and B cells again today. So this is how the three lines of defense is given by your immune system in the body. Is it clear? Okay. Now, if I see the adaptive immunity, now let's see adaptive immunity. This one, adaptive could be T cell mediated and B cell mediated, right? And T cell mediated is called the cellular, because you know that T cells directly get attached to the antigens. So T cells get attached to the antigens directly and try to destroy the antigens. That's why called the cellular immunity. And B cells produce antibodies, secrete antibodies, and then that antibodies will destroy the antigens. So that is called humoral, because humoral indicates humor, fluid. Humoral, aqueous humor, vitreous humor inside the eyeball, right? So fluid, humoral. So cellular and humoral. Cellular given by the T lymphocytes or T cells, and humoral given by B lymphocytes or B cells. And cellular work directly by phagocytosis or direct attachment, and B cells, humoral, fight by producing and secreting antibodies. Make sense? So this one, T cells mainly by phagocytosis. Okay. Now the humoral. This one, if we see the humoral immunity, it could be two types: active humoral and passive humoral. Okay, so two types of humoral. We'll talk about all these. And active humoral and passive humoral both could be natural and artificial. This one, passive, could be also natural and artificial. So this is the entire classification of the immunity. So immunity could be two types: innate and adaptive or acquired, right? Innate gives first line of defense and second line of defense. Adaptive gives the third line of defense. And adaptive could be T cell mediated or B cell mediated. And T cell is cellular and B cell humoral. Okay, and humoral, that means the secretion of antibodies could be active and passive humoral, and both active and passive could be natural and artificial. Make sense? So that's the interclassification of the immunity. You see, cellular, humoral, right? Then active and passive, right? Humoral is two types, active and passive. Then both could be natural and artificial. So let's see. So now you know that the innate everybody get by birth, like skin, the lining, mucous membrane, inner lining of your respiratory and GI tract, and the mucus lining. So it is non-specific, because you know that skin and the lining of your GI tract and respiratory tract will try to stop everything, right? Your skin doesn't have any specific selectivity. It is like a membrane, so it will try to stop everything. So non-specific. Your skin cannot detect what type of antigen is that. It cannot separate this one bacteria from another. It is just like a barrier that will try to stop everything. So non-specific. And present at birth. Adaptive or acquired is highly specific, as I mentioned. If you get swine flu virus, only your body will have that immunity against that, right? But someone did not get exposed to swine flu, he won't have that immunity. So highly specific, based on the antigen you got exposed to, your body produces that. Okay, and that's why the response varies, okay, and develops after birth, okay, throughout the life in response to the antigen. So those are the main differences between the innate and adaptive. I already talked about the first line, second line, and third line of defenses. So physical barrier, skin, mucous membrane, that means the lining of your GI tract and respiratory tract, right? Mucous membrane. So this is first line of innate, right? So first line. And phagocytes, the macrophages, right, in the tissue, and you have some natural killer cells. Those natural killer cells, they can detect some antigens and kill them. These are present in the tissue. So tissue phagocytes, tissue natural killer cells, the protein, you remember, secreted in the tissue, chemicals, interferons, complements, cytokines, those also belong to second line. So all these belong to second line of defense, okay, and this one is the first line of defense. Okay, we'll talk about the interferons, complements, inflammation, and fever. Just know that all these belong to the innate immunity, which is non-specific and gives first line and second lines of defense. And adaptive immunity is highly specific and gives the third line of defense. So your skin, you see, this is the structure of the skin. Skin is a barrier, right? It covers the whole body, and if any antigen tries to enter into your body, your skin will first try to stop that, right? So it belongs to the first line of defense. Skin has the stratified squamous. You must remember, squamous are the flat cells and stratified indicates many layers, right? So many layers of flat cells, stratified squamous here, and these cells are filled with keratin, a protein that makes this layer uh, 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 very, uh, uh, very uh, waterproof and strong. So the antigen will not be able to pass through the layer. Makes sense? So keratinized cells, these cells are filled with that protein called keratin that makes the layer very strong and waterproof. So the antigen will not be able to pass through it. Also, in your skin, cells have desmosomes. Desmosomes are the uh, protein structures. Those are called the junctions, and these are very strong junctions, so the antigen won't be able to pass through these junctions. So the antigen will not be able to invade. Makes sense? So these two things in your skin would not let the antigen pass through the skin. Also, skin has sebaceous gland, and sebaceous gland secretes sebum. Sebum is the secretion, and that sebum has antibacterial chemicals or agents. So bactericidal action of sebum would destroy the antigens. Okay, so these are three important things in your skin that would try to stop and destroy the antigen. However, if the antigen somehow manages to pass through this skin and enter into the tissue, underlying tissue, then second line of defense will be activated. The phagocytic cells, right, and the protein chemicals, interferons and complements, those will be activated to destroy the antigen. Okay. So this is the respiratory tract, and you see the inner lining. This is the inner lining and secretion of mucus. So the antigen will not be able to pass through the inner lining of your GI tract and gastrointestinal tract. Okay. Now, if somehow the antigen breaks the skin or the mucous membrane, that means somehow can pass through the first line of defense, then in your tissue you have phagocytic cells as well as natural killer cells that I mentioned, and natural killer cells are specifically very effective to destroy certain, recognize and destroy bacteria, foreign cells from outside, virus infected cells and cancer cells. That means your own abnormal body cells can be detected by the natural killer cells. These cannot be easily detected by your immune system. A normal immune system normally ignores, but the natural killer cell will be able to detect and destroy it. Interferons also belong to the second line of defense. What are the interferons? Interferons are protein chemicals from the tissue and secreted from the viral infected cells or activated lymphocytes and macrophages. So if this is your body cell and these are the other cells around it. So if this cell got infected by virus, okay, so what will happen? This viral infected cell will secrete a chemical that is called the interferon. Okay, so interferon, there are different types of interferons, so that is one. And also the presence of virus or antigen will activate the macrophages, right, phagocytic cells, as well as the lymphocytes. So macrophage or lymphocyte will be also activated and will secrete interferon. So viral infected cell or activated macrophage or lymphocyte would secrete that chemical interferon, which is very important to alert these healthy nearby cells. So the interferon will alert these healthy cells and tell them to produce uh, antiviral proteins, okay, and that antiviral proteins will protect these healthy nearby healthy cells. Make sense? And that antiviral protein would not stop the invasion of the virus, but would stop the replication or division or multiplication of the virus. Okay, and that's how the healthy tissue or cells will be protected by the interference. Make sense? Okay, so interferon complement. There are more than 30 different types of complement proteins in your tissue. So complements also belong to the second line of defense, and complements are activated by two different ways. One is called the classical pathway, another is called the alternative pathway. Okay, so the complements over 30 different types of proteins, and they uh, belong to the complement system. Okay, and why they are called complements? Because they complement the function of the antibodies. You know that antibodies fight against the microorganisms or antigens, right? Antibodies fight against the antigens, and these antibodies are complemented by these proteins, okay, or helped by these proteins. So antibodies functions are enhanced uh, or increased by the complement proteins. And how the complements work or be activated by two pathways, okay, classical and alternative. Classical is more effective and rapid, okay, activation method. So we'll see how they work. Alternative pathway work through the proteins. Those are called propertins, okay, and classical pathway complements, they will bind with the antibodies on the plasma membrane of the bacteria or antigen. So let's see here. You see the alternative pathway of complement function and classical pathway of complement function. So in case of classical, which is rapid and more effective, you see this is the wall of a bacterial cell wall. So this is the bacteria. If you just see a part of the bacterial wall, this is the wall, and these Y-shaped protein structures are the antibodies. Okay, so antibodies will first get attached to the plasma or cell membrane of the bacteria, and then the complement proteins will get attached to these antibodies. So these are the antibodies, and this blue one is the complement protein. Okay, when this complement protein will be attached to the antibodies, this complement protein will activate other complement proteins. There are many different types, right? So these complement proteins will be activated, and the activated complement protein, this one, will get attached to the cell membrane of the bacteria and will make hole in the wall. So you see, activated complement protein got attached to the cell membrane of the bacteria, and that is the classical pathway. So this activated complement will make hole in the wall. So the fluid will get into the cell and will cause the lysis or breakdown of that bacteria. So by making holes or pores in the bacterial wall, it will destroy the bacteria. Makes sense? That is called the cell lysis or bacterial lysis. Okay, multiple pores or holes you see here. And also some complements will get attached to the surface, like this is the bacteria, and these are the activated complement proteins, and will make this bacteria more tasty for the phagocytic cells to eat it up. So this is a phagocytic cell, for example your macrophage or any white blood cell. So this is a phagocytic cell, and this is the bacteria, and when these complements, activated complements, will get attached to the surface, that bacteria will be more, delicious. So this uh, phagocytic cell or macrophage would love to eat that bacteria and destroy it. Makes sense? So that is enhanced phagocytosis, and this process is called opsonization. Opsonization. The term opsonin is side dish. After you eat dinner, you have a side dish. So that is the term opsonin, and these are the opsonins complements, and will make it delicious for the phagocytic cells. Okay, and also when the complements get attached to the surface of the antigen, for example bacteria, that will activate the mast cells to release histamine. So histamine will be released from the actually the basophils, one type of white blood cells. Basophils will release histamine, and that will also help in immunity by inflammation. Okay, so you see, this is the classical pathway, and alternative pathway, you see, the properdin protein, complement protein, would get attached to the bacterial cell wall. So that's the activated complement protein. So properdin will produce activated complement protein, and the same three ways. Once the activated complement proteins are attached to the cell membrane, bacterial cell membrane, they will destroy the bacteria by these three processes. One is making holes or pores that would cause the lysis or breakdown. Another is enhancing phagocytosis, and another is by releasing histamine from the basophils, and that will increase inflammation. And when the inflammation occurs, the immune function in that area is increased. Okay, now why inflammation is good? although we get bothered by inflammation, but inflammation actually helps the response when your tissue uh, gets infection. Inflammation occurs. And what are the signs of inflammation? Swelling, redness, right? In that area at the site of inflammation, if you touch the site of inflammation, you will feel the area is slightly warm because more blood flows. Why that area looks red? Because more blood flows, blood vessels dilate, so blood flow increases. Increase blood circulation or blood flow. Makes sense? So that area would look red. And swelling, because some fluid will come out from the blood and that will accumulate in that area at the site of inflammation, and that will cause swelling. Heat, when more blood flows in that area, blood is slightly warmer than your body, so that area will be slightly warmer, slightly warm. And the chemicals, those are released at the site of inflammation from the tissue, will cause pain. For example, prostaglandin, right? Bradykinin, those chemicals, histamine, heparin, those are released, and because of those chemicals, you see these changes at the site of inflammation. So, which chemicals? Histamine, right? Heparin, prostaglandins, different types, bradykinin. Makes sense? So these chemicals will make these changes at the site of inflammation, and these chemicals are released from the cells in the tissue. Okay, so chemical changes. Fever also helps in immunity. When the body temperature increases above 37.2 degrees Celsius, that's 99 degree Fahrenheit. If the temperature is above that, that is considered as fever, right? And what causes fever? Certain chemicals that are released in the body when you get infection, antigen enters into your body, you often gets fever because your body releases pyrogens. Okay, pyrogens are fever inducing proteins. Okay, and that proteins changes the function of the hypothalamus in the brain, and the hypothalamus is the center that regulates the body temperature, regulates body temperature. Okay, so hypothalamus function is changed, so the body temperature cannot be controlled or regulated, so that may cause increase of body temperature, and that's the fever. Okay, now why fever is also helpful? Both inflammation and fever are actually good, because as you remember, when inflammation occurs, more blood flows to that area, right? And also, when fever or body temperature increases, the metabolism increases in your body, and when more blood flows to that site of inflammation, if this is the site of inflammation, these are the pathogens or antigens, right? Bacteria or virus. And when more blood will flow, more white blood cells will go to that area, right, with the blood, and white blood cells fight the antigens, so more antigens will be destroyed. So that is helped by inflammation. Make sense? Fever increases the metabolism in the tissue here. So if metabolism increases in this area, then what will happen? The antibodies and immune function will also be enhanced. So fever increases metabolism, and that increases the immune function or immunity, enhances the immunity. Okay, and inflammation increases the blood flow or circulation, blood flow, and that would increase the movement of white blood cells. White blood cells will move to that site, and white blood cells will destroy the antigens. Make sense? So both are beneficial for our immune function to protect your body. Okay, now you know that the adaptive immunity, also called acquired, provides the third line of defense I already mentioned, and given by T lymphocytes and B lymphocytes, right? And P lymphocytes mainly work by direct attachment with the antigen and causes phagocytosis. That's why called cellular. And B producing antibodies. That's why called humoral. I explained that before. So here you see the adaptive immunity. Cell mediated or T cell mediated, and antibody mediated or B cell mediated immunity. Okay, and T cells directly get attached to the antigens and would destroy them usually, but also can release chemicals that would destroy. So either by direct physical attachment or by releasing chemicals, T cells. And B cells by producing antibodies. So B cell mediated is also called humoral, right? Because secretion of antibody, humoral. Okay. So here these are the main phagocytic cells. Neutrophils are one type of white blood cells, and eosinophils. These are the main phagocytes or phagocytic cells in your blood, white blood cells, these two types of white blood cells, and macrophages. Macrophages present in the tissue, and macrophages could be fixed in the tissue, present in one location, want to move. Another could be free type, free flowing or circulating macrophages. Okay, they can move. Okay, now different types of T cells and B cells. T cells could be four different types: cytotoxic T cell, helper T cell, regulatory T cell, and memory T cell. Okay, and B cells could be plasma cells and memory cells. Now cytotoxic T cells, its name, you see, cyto means cell, toxic means harmful, right? So these type of T cells would attack the cells and destroy them. Cells means the bacterial cell, or any foreign cells, or from another person's body, tissue transplantation or cell transplantation. Those foreign cells will be considered as enemies, right? Or the cancer cells or tumor cells or abnormal cells in your body. So cytotoxic T cells will attack those cells and destroy them. That's why called cytotoxic. So what happens normally, this is a cytotoxic T cell, and this is, for example, bacteria. Okay, cytotoxic T cells would release when the bacteria will come close, or abnormal cancer or tumor cell will come close to it. It will release a chemical that is called perforin. Okay, and these perforin molecules, protein molecules, will get attached to the cell membrane of the bacteria and or abnormal cell, and will make holes or pores. So perforin will make pores. So many pores will be formed, and then what will happen? This cytotoxic T cell now secrete an enzyme that is called granzyme. Okay, and that granzyme molecules, protein molecules, will enter through those pores or holes into the abnormal cell or bacterial cell, and will stop metabolism, will stop cellular functions or metabolism. Okay, and the cell will die. So I'm repeating again, the cytotoxic T cell, this is the cytotoxic T cell. When the abnormal cell or bacteria antigen will come close to it, it will release perforin, and that perforin will get attached to the cell membrane and will make many pores, and then the cytotoxic T cell will secrete granzyme, and the granzyme molecules will enter into the bacteria or abnormal cell through the pores and will stop its metabolism or cellular functions, so it will die. Okay, and cytotoxic T cells also known as natural killer cell. So some natural killer cells are cytotoxic T cells. Helper T cell, how the helper T cells work? This is interesting. This is a helper T cell. Helper T cell. Helper T cell is inactive normally, and it needs to be activated. Who activates the helper T cell? The antigen presenting cell in your body. This is another cell. This is called the antigen presenting cell. Its name is telling you what it will do. This is the antigen, bacteria or abnormal cell. So this is the antigen. So when the antigen will come close to the antigen presenting cell, it will hold the antigen and will present to the helper T cell. So that's why it is called the antigen presenting cell. Okay, we'll hold it and bring it to the helper T cell, and helper T cell will then secrete cytokines, proteins. Cytokines could be interferons or complements. All those belong to proteins, belong to cytokines. Okay, and the cytokines will then do what? Will activate both cellular and humoral immunity. That means will activate the T cells as well as the B cells. And when the T cells will be activated, they will destroy. When B cells will be activated, they will secrete antibody and destroy the antigens, right? So that's how the helper T cell works, to destroy the antigens. Regulatory T cells are also called suppressor T cells, and this type of T cells are very important in your body because it controls the immune function. If your immune system tends to work too much, over function, then it will slow it down. Make sense? So that's why it is called regulatory T cells. Regulatory T cells will tell the immune system of your body not to work too much. Now, if regulatory T cells fail to work, then what will happen? Your immune system of your body will overact. Even if you get a small antigen, a small amount, your body will act more than it needs. So what will happen? That may cause problem. when your immune system will be hyperactive, that can attack your healthy cells too, right? And that would cause autoimmunity. Autoimmunity. Normally, your immune system should attack the abnormal cells, but if the function is uncontrolled, then that might cause autoimmunity. That means your healthy cells could also be attacked by the immune system, and that could be a problem. There are many autoimmune diseases. One good example could be rheumatoid arthritis. There's tissue in your joints are affected by the immune system, a healthy tissue. So that causes inflammation in the joints. Memory T cells are also known as experienced T cells. That means the T cells are experienced. Experienced to what? Experienced to the antigen before. So antigen experienced cells. Antigen experienced cell. What does it mean? If you get, for example, COVID first time, then some T cells will store that information as memory. Make sense? So some T cells will store the information about COVID antigens or virus in your body, and after you get recovered, when the second time same type of antigen like COVID would try to invade into your body, since these T cells already have memory, these are antigen experienced cells, they will quickly act to destroy the COVID virus when they will try to invade second time or third time. So your body is well prepared because of the memory T cells. Make sense? And that we all know that if you get sick once, the chance of getting sick second time or third time is less because of the memory cells. Okay, now B cells, many B cells are converted to plasma cells, I mentioned before in last class, and plasma cells can secrete plenty of antibodies because we know B cells act by secreting antibodies, right? Memory B cells also, B cells could be memory type B cells, that means can store the memory of previous invasion. So those are different types of T and B cells. Okay. Here you see I already actually explained how the cytotoxic T cells or natural killer cells work. So this is the cytotoxic cell, and this is the bacteria or abnormal cell like cancer cell or tumor cell. You see when they come close to each other, the cytotoxic T cell or natural killer cell would release perforins. So you see perforin molecules will be released and will get attached to the membrane to make holes or pores. So granzymes will enter and destroy them. Okay, so summary of innate immunity. I already explained all these. This one gives the first line of defense, and these belong to the second line of defense, right? Phagocytic cells, two types of white blood cells I mentioned, neutrophils and eosinophils, and the macrophages, fixed and free. So they are the main phagocytes or phagocytic cells. Then immunosurveillance cells or immune surveillance cells are the natural killer cells, right? Destroy abnormal cells. Interferons will be released from the viral infected cells to alert the adjacent healthy cells, right? So viral multiplication or proliferation will be stopped. Then complement proteins, more than 30 different types. Some of them work through classical pathways, some of them work through the another pathway. So we talked about both pathways, right? Classical pathway is more active, and alternative pathway is less active and slow. However, both would do three things: make holes, and that could cause the lysis of the bacteria or antigen, opsonization, and increasing inflammation by releasing histamine. Okay, so we already talked about all these things: inflammation and fever. Okay, now adaptive immunity, B cell mediated immunity uh, is one type of adaptive, and T cell mediated another type of adaptive, right? And I mentioned that the adaptive could be active or passive, and both active and passive could be naturally acquired and artificially acquired. That means natural and artificial. Okay, so let's do here. Adaptive could be active or passive. Now just think about the names. Active adaptive immunity is triggered or activated when an antigen enters into your body. That means antigen, it can also be produced in your body, for example abnormal cells, right, or can come from outside, bacteria or virus or pathogens. Whatever, the antigens would trigger the active immunity. Make sense? So antigen is needed to trigger the active immunity. So in response to that antigen, your body will produce immunity. Make sense? So that's why it is active. Body is actively producing in response to antigen. Passive immunity your body gets not by the presence of antigen but antibody. So your body doesn't need um, the antigen to produce passive immunity. Okay, it is given by antibody without the presence of antigen. Now active could be natural and artificial, right? Natural, artificial. Now good example of natural is what? If any bacteria or virus enters into your body, we all know that body produces antibody, especially after COVID we know more, right? So your body will produce immunity when antigen antigen enters or can be produced in your body. Then that is naturally occurred, because you know that bacteria, virus, those are coming from the nature. And artificial is vaccine. Good example is what? Vaccine. You know that vaccine is produced in the labs, right? So it is artificial. Vaccines are actually uh, highly attenuated or inactivated antigens. So vaccines are actually antigens, but we are producing in the lab by making the antigens um, very weak, but they should be able to trigger the response, immune response. So we highly attenuate or make the antigens weak and then inject it to the body. So why we need to make the antigens weak? Because they cannot cause disease, but they are enough to trigger the immune system in your body. Makes sense? So that is the artificial. Passive could be also natural and artificial. Passive, your body gets antibody directly. Okay, your body is not producing antibody in response to antigen. What could be a good example of natural passive? Baby gets milk from the mother's body, right? And that milk, mother's milk, has a lot of antibodies. So baby is ready-madely getting the antibody from outside. So baby's body doesn't need to produce, right? So that is naturally occurred antibody in baby's body, getting the antibodies without antigen. And artificial, if someone gets HIV, AIDS, that HIV um, uh, or AIDS virus do what? Destroy the T cells. That means destroy the immune system or function of your body. So that person's body's immune system is almost destroyed. So what you have to do, since that person's body cannot produce antibody or immunity, you have to give antibodies directly from outside. Okay, so we make the antibodies in the lab and we'll inject into that person's body, because that person's body cannot produce antibody, so we have to give from outside, right? So in case of passive, we give antibodies from outside. Babies get from mother's milk, and AIDS patient get from outside, as injected form. So that is artificial, because we make in the labs. Okay, so this is the classification: active, passive, natural, artificial, natural, artificial. Okay. Major histocompatibility complex. Major histocompatibility complex, or MHC, are the proteins, and those proteins actually help to activate the helper T cells. You remember the antigen presenting cells. The antigen presenting cells have the proteins called the major histocompatibility complex, and that helps to activate the T cells. Okay. Antibodies. Now antibodies are produced in response to antigen. So when antigen enters or produced in your body, in response to that your body produces antibodies, right? And if you see the antibodies, 5 different types of antibodies are very common. We'll talk about that. But if you see the structure of an antibody, antibody is usually Y or T shaped. So this is an antibody, you see, this is Y shaped antibody, right? And it could be also T shaped. Now antibodies are protein structures formed by the protein chains. So how many chains you have here? You see 1, 2, 3, 4, right? And these 2 are heavy chains because they are big, longer, and these 2 are light chains. Make sense? And now if you see more carefully, both heavy and light chains have 2 parts. This part, the end part, are variable parts. Okay, and the bottom parts are fixed or constant parts. Okay, makes sense? So each polypeptide chain or protein chain has variable part and fixed part. Fixed, variable. Makes sense? And this part, usually the variable part changes based on the shape of antigen. It's like, sometimes we use the screwdrivers that have different sized top part, but the shaft part is fixed, right? So this is the shaft part. Here in your car you keep that tools. This part you can change. You can make it small one, attach here, or change with big one, but this part is fixed. So this is the fixed part or constant part, and this is the variable part, and this part you use, right, and change to unscrew something. So similarly, the variable part can be changed depending on the shape of the antigen. For example, this is an antigen and these are the harmful sites of this antigen. So the Y-shaped antibody will be like this. You see, this is the antibody and this is the variable part, right? Will get attached to the harmful site of the antigen to mask it or block it. Now, if the antigen has smaller harmful sites, then this variable part will be changed, but this part will remain same. So this part will be smaller, the variable part, and the other part will remain same, okay, the shaft part. So that's why we have variable part based on the size of the antigen binding sites. So this is an antibody, and you can see the heavy chains and light chains, right, and antigen binding sites here, variable segment and constant segment. Okay. For the antibodies to work or function, antigen antibody complex must be formed. That means the antibodies must be attached to the antigens to form the complex, and when the complex is formed, that means antigen antibody binding takes place. Then a number of functions are activated to destroy the antigen. Now, as I said, that 5 different types of antibodies are the main types: immunoglobulin G, Ig, or immunoglobulin E, immunoglobulin D, immunoglobulin N, M, and immunoglobulin A. So immunoglobulins, you remember, globulins, just know that are proteins, and immuno, that means the proteins that give immunity. These are immunoglobulins. Okay, so these are proteins and different types of proteins, and these are 5 most common type of antibodies. And antibodies, when antigen antibody complex is formed, then it can cause a number of things to inactivate or destroy the antigens. Neutralization, that I showed you, that if this is the antigen, antigen has harmful sites, like when you fight, your hands and legs are the harmful sites, right, for your enemy. So these are the harmful sites, and antibody, the variable part, would block that. Make sense? So this is the Y-shaped antibody. Okay, so this antigen will not be able to cause harm. That is neutralization. Okay, another is adhesion and activating complements. You remember the classical pathway. The antibodies first get attached to the plasma membrane of the bacteria, then the complement protein gets attached to it, and that activate other complement proteins. Also, by producing inflammation. Inflammation helps in immunity, right? Opsonization, I explained that, making the antigen more tasty or like side dish. and phagocytosis. So all these things can be activated once the antigen antibody complex is formed. Okay, so here you see all those mechanisms, how the antigen antibody complex works. Okay, so antibody, here you see these are the harmful sites are masked by neutralization. This is the antibody, right? So this is the these are the ways how the antibody works. Okay, so that's all about the immunity. So first thing, you make sure you know how to classify the entire immunity that I showed you um, at the beginning of my talk. Okay, thank you.